Hey everyone, welcome to The Explainer. Today, we're jumping right into the fascinating, incredibly dynamic world of transcriptomics to uncover exactly how data science is transforming modern healthcare. If you've ever wondered how scientists actually look inside a living cell to see what it's doing in real time, well, you're in the exact right place.
So, we're going to start with the ultimate biological mystery to really get things going. Think about this for a second. Almost every single cell in your body contains the exact same genetic blueprint, right? The DNA in your eye is literally identical to the DNA in your liver. So, how do identical genetic blueprints build entirely different cellular machines? I mean, how does a heart cell know it needs to beat, while a brain cell knows to fire electrical signals? The answer, surprisingly, doesn't actually lie in the DNA itself, but in how that DNA is used.
Let's dive right into Section 1. What is the transcriptome?
To solve our mystery, we have to move past basic genomics, which just looks at the static, unchanging DNA, and step into the active, bustling construction site of the cell. To make sense of this, let's use an analogy. Think of your DNA as a master reference book. It's incredibly valuable, right? So it stays locked safely inside a library, which is the cell's nucleus. It never leaves. But the cell is like a busy kitchen that needs to cook up proteins to survive. So what does it do? It makes temporary, active photocopies of specific recipes from that master book. These photocopies are called messenger RNA, or mRNA. The transcriptome, therefore, is the entire stack of these temporary photocopies that the cell is currently using as recipes in the kitchen at any exact given moment.
It's really the dynamic approximation of life. Picture a static, unmoving DNA double helix on one hand, and contrast that with an active, bubbling, changing cell on the other. Unlike your static genome, which is exactly the same whether you're sleeping, running, healthy, or sick, the transcriptome is incredibly dynamic. It is constantly changing. The cell is continuously creating and destroying these RNA photocopies to approximate the exact expression of its genes based on real-time conditions. So if the cell is stressed, it prints different recipes. If it's dividing, it prints different recipes.
Moving right along to Section 2, why gene expression matters.
Okay, so why do we actually care about these molecular photocopies in the real world? Why go through all the trouble of measuring them? Well, the identity and health of our tissues are largely defined by which recipes are turned on or off. By measuring these RNA molecules, we can see the specific types and amounts of proteins a cell plans to make, and this correlates directly with biological changes across different conditions. For example, a healthy lung cell is going to have a very specific stack of RNA photocopies, but a lung cell battling a virus, its stack of photocopies will look completely different because it's actively expressing different genes to fight off the infection. Measuring these changes is absolutely crucial for understanding the true biological state of a patient.
Which brings us to Section 3, how RNA sequencing works.
So the big question is, how do we actually measure millions of these tiny, invisible messages flying around a cell at any given second? The answer is a real game changer, a technology called RNA sequencing, or RNA-seq. By using massive parallel sequencing, RNA-seq allows us to simultaneously read all these active recipes at the exact same time. It's kind of like taking millions of shredded documents and reading them all in one go. This lets us compare two different samples, say a healthy cell and a cancer cell, and see exactly which genes are turned on or off. We call this tracking differential expression. On top of that, RNA-seq actually allows scientists to reconstruct entire transcripts from scratch and detect specific variations of genes, which we call isoforms. Think of isoforms like slightly different variations of the exact same recipe. Maybe one calls for a pinch of salt and the other calls for a dash of sugar, but they came from the same master page.
Let's move to Section 4, choosing the right tools. Just like any big data project, getting an accurate picture of the transcriptome requires selecting the correct sequencing hardware. And in data science, your analysis is literally only as good as the machine gathering your data. Let's compare the two heavyweights in the field right now. First, we have Illumina, which uses short reads. This is the industry standard, it's highly affordable, and very accurate. To put it simply, using Illumina is like flawlessly shredding a document into tiny confetti and then using a massive computer to piece it perfectly back together. On the flip side, we have nanopore sequencing. This is an incredibly portable technology that allows for real-time and direct RNA sequencing. You could literally take it into the jungle to sequence a virus. But it definitely has drawbacks. Nanopore is more like reading a continuous, long scroll that might occasionally have some smudged ink. It suffers from lower coverage and highly replicable errors where the machine simply misreads the sequence.
Next up, Section 5, designing flawless experiments. Let's shift gears to the data science side and see how this builds into designing flawless experiments that give us numbers we can truly trust in healthcare. The first major hurdle in experimental design is figuring out your sequencing depth. Imagine taking a magnifying glass to try and find a very specific, rare recipe hidden in a massive pile of millions of photocopies. How closely do we need to look? Well, the number of reads or individual pieces of RNA we need to sequence is determined by a few things: the least abundant RNA species we care about finding, the overall size of the transcriptome we're studying, and the specific biological question we are trying to answer.
The size of the genome actually drastically changes our data requirements. For instance, a small bacterial genome might only require 5 million reads to successfully map out differential gene expression. But exploring a large human genome, that requires exponentially more sequencing depth, up to 20 to 50 million reads just to reliably compare active genes. Simply put, the bigger the library, the more reads you need to ensure you don't miss the rare, quiet genes that might actually be the ones causing a disease.
But listen to this. Even with millions of reads, your data can still be totally ruined by what data scientists call nuisance factors. These are variables you really don't care about, like, say, the temperature of the room or the specific technician handling the sample, that might accidentally influence the outcome. To ensure our data represents true biological differences rather than just random noise, we have to employ rigorous statistical safeguards. Two of the most important are randomization and blocking. Picture a handful of scattered dice representing randomization. We literally use random choices for these nuisance factors so they don't skew one specific group. Now picture neatly organized test tubes representing blocking. Blocking is when you group data to keep an annoyance factor constant. By keeping the annoyance constant in a block, you can cleanly assess the real biological variance you are actually interested in.
Which brings us to the golden rule of experimental design for RNA-seq. Block what you can, randomize what you cannot. I want you to really let that sink in. If you memorize just one phrase from this entire explainer to ensure your healthcare data is accurate and free from bias, let it be this. This is exactly how data scientists guarantee that the gene changes they discover are real, and not just a glitch in the experiment.
So, bringing it all back together, why is measuring the transcriptome so vital for healthcare? Ultimately, tracking this dynamic, ever-changing construction site inside the cell helps us understand exactly how gene regulation varies across different cell types, different health conditions, and completely diverse genetic backgrounds. It bridges the gap between our static DNA and the living, breathing reality of human biology. This is the very foundation that is unlocking the future of targeted, personalized medicine. And it leaves us with a pretty thrilling question to ponder. If we can now read the changing instructions of life in real time, what previously unsolvable diseases are we about to cure next? Thank you so much for joining me on this explainer. Keep asking questions, and I'll catch you on the next one.